To understand the natural world, humans have always needed to organize it. Faced with millions of different organisms, we have an innate drive to sort the chaos of life into a logical, manageable system. The earliest recorded attempt came from Aristotle. He categorized life using simple, observable traits, dividing animals into those with red blood and those that were bloodless, like insects and marine invertebrates. By the 18th century, Carolus Linnaeus formalized a broader binary system that would dominate science for hundreds of years. He split all living things into exactly two categories, kingdom Plantae and kingdom Animalia. The criteria were strict and based entirely on outward appearance. If an organism was green, rooted to the ground, and had a rigid cell wall, it was a plant. If it moved around and consumed food to survive, it was classified as an animal. For a world observed only with the naked eye, this binary made intuitive sense. A tree is clearly a plant, a dog is clearly an animal. But as glass lenses improved and we looked closer, this neat division began to fracture. The invention of the microscope revealed a hidden universe of organisms that completely defied Linnaeus's definitions. Biologists suddenly had to classify things that blurred the lines between plant and animal. Take fungi, for example. A mushroom grows from the ground and appears rooted like a plant. But fungi do not photosynthesize. They are heterotrophs, organisms that consume organic matter for energy, meaning they essentially eat their surroundings, much like animals do. Then there is the euglenoid, a microscopic freshwater organism. It is mixotrophic, meaning it uses a combination of nutritional strategies. It has chloroplasts and photosynthesizes in the sunlight, acting like a plant. But in the dark, it becomes a predator, swimming around to hunt smaller organisms. The most glaring problem was bacteria. Because bacteria possess a cell wall, early taxonomists shoved them into the plant kingdom. Yet bacteria are prokaryotes, meaning they lack a true nucleus to hold their genetic material, making their internal cellular structure wildly different from an oak tree. These anomalies piled up. Grouping organisms by outward traits, like the presence of a cell wall, grouped highly complex multicellular organisms with primitive single-celled microbes. The science of taxonomy required a much deeper set of biological criteria. In 1969, an ecologist named R.H. Whittaker proposed a solution that reorganized our understanding of life. He introduced the five-kingdom classification system. To avoid the traps of the old binary, Whittaker built his system on 5 specific pillars: cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. Phylogenetic relationships refer to the evolutionary history and ancestral lines connecting different species. This logic tree shows the crucial dividing line: cell structure. He split life into prokaryotes, lacking a nucleus, and eukaryotes, which have a defined nucleus. All prokaryotes formed a single category, kingdom Monera, the most ancient cellular survivors on Earth. Within Monera, we find immense diversity. There are Archaebacteria, extremophiles that thrive in boiling hot springs and highly acidic salt marshes, and there are Eubacteria, the true bacteria that exist almost everywhere else in the environment. By prioritizing cellular architecture over outward appearance, Whittaker isolated the oldest, most distinct branch on the tree of life, finally separating bacteria from plants. With the prokaryotes isolated, Whittaker turned to the eukaryotes. Here he applied his second filter, body organization, cleanly separating single-celled life from complex multicellular organisms. All unicellular eukaryotes were gathered into kingdom Protista. This kingdom serves as a diverse collection of single-celled organisms, including silica-shelled diatoms and marine dinoflagellates, which have pigments that can cause massive, glowing red tides in the ocean. His final filter for multicellular eukaryotes was mode of nutrition. The saprophytic path, absorbing nutrients from decaying matter using chitin lulls, leads to kingdom fungi. Autotrophic producers using sunlight become kingdom Plantae. Heterotrophic consumers that ingest organisms form kingdom Animalia. These specific nutritional strategies dictate the functional architecture of every ecosystem, linking producers, consumers, and decomposers into a continuous cycle of energy. When you look at a vibrant, high-diversity ecosystem like a dense forest floor, Whittaker's classification system ceases to be an abstract chart. It maps directly onto the physical world. Invisible to the naked eye, Kingdom Monera saturates the soil, fixing nitrogen and cycling chemicals, while single-celled members of Kingdom Protista navigate the stagnant pools of rainwater. By stripping away superficial appearances and focusing on how cells are built and how they survive, we mapped the true continuum of life, linking a microscopic bacterium in a boiling spring directly to the complexity of a human being.